Okay, thank you very much. Thank you for having me, Demir. And it was a pleasure meeting with all of you. I missed some of the early talks, but I enjoyed the discussion group. And so like most people do at conferences, went and added some more things to my slides just to kind of make sure I covered some things that I wanted to talk to for this audience. So as mentioned, I bring you greetings from Georgia Institute of Technology and Emory University, where I run the Platt Lab for Repair, Regeneration, and Remodeling. We do several different things in my lab, but one of our major research interests is actually sickle cell disease. And so of all the things that we do, come on and play for me, baby. There you go. Ooh. Fine. So we study sickle cell disease, again, in the large arteries. And it is traditionally studied in the small vessels, which we'll talk about why we look at the large arteries. We also study how people with HIV have increased risk of heart attacks and strokes. And these mechanisms are not really understood, whether it's inflammation due to the virus or the drugs themselves. This has become a new research interest in our group. Plus, we do some early cancer detection and predictive medicine. And you'll see how that all plays into our work. But I say that we're the lab for remodeling. So what is tissue remodeling? So we think of tissue remodeling as how you go from this open, happy vessel or this healthy tissue to some disease state. So this here is actually a coronary artery that you can imagine blood is coming out of the board. But then when you get to this atherosclerotic plaque, as you can see, there's lots of protein synthesis, cell infiltration. But some of the initial steps are actually protein degradation. And so we really study a lot of protein degradation in tissues. So again, the focus of this talk will be actually about sickle cell disease, and there are a few talks after me that will describe it, so I'll just give the basic overview. It's a single point mutation. It's this amazing disease that just a single point mutation, I call it the singularity of sickle cell, that leads to this multifactorial disease. So one nucleotide changes from an A to a T, which changes one amino acid from a glutamic acid to a valine that's only in one protein, hemoglobin, that's only in one type of cell, red blood cells. But you have this multifactorial systemic damage that leads to much shorter life expectancies. Most recent, it's 36 for males, 42 for females. And it's due to this sickling of this hemoglobin, which I'll get into on the next slide. But in the United States, 1 in 400 African Americans has this disease, and 1 in 12 actually have sickle trait, or the recessive form. So it's actually quite common, or relatively common among genetic disorders. But the molecular origin of sickle cell disease, it was the first molecular disease identified. And again, it's from this hemoglobin that's in your red blood cells that carries oxygen and carbon dioxide. Under low oxygen conditions, it exposes that valine, which is a hydrophobic amino acid, that then causes all of those to start to cluster together to sequester that hydrophobic moiety. And you get this very highly ordered hemoglobin polymer, which our next speaker will give lots of details on, so I'll just gloss over it. But that actually ends up deforming the red blood cell. And again, if you remember, the red blood cell does not have a nucleus, which is the major rigid body in cells, allowing this complete deformation. So a red blood cell in a person with sickle cell disease actually only survives about 9 to 10 days. And in a normal human or a person without sickle, it's about 120 days, which leads to this anemia that develops. But what most people think about with sickle cell is actually the vaso-occlusion that occurs. So when you get this hemoglobin sickle polymer forming inside of these red cells, in the post-capillary venules, you can actually get vaso-occlusion, which will then block the blood flow, become very painful, lead to ischemia, and then, of course, once it resolves itself, now you have ischemia-reperfusion injuries. But this is extremely painful, and this is constantly happening. As you can imagine, they're constantly entering low oxygen environments in the blood. But what drew me, really especially, pulled my heartstrings was that I actually found out that children with sickle cell disease are at increased risk for strokes. So normally you won't have a stroke till 50, 60, 70, 80, a lifetime of McDonald's and laying on your butt, right? But these are children. So 11% of children with sickle cell will have a stroke by the age of 20. And this is actually a paralytic stroke when they describe this. But the greatest risk is between the ages of 3 and 5 years old. So you can see this angiogram here, the blood is fully perfused, but this is actually the brain of a 3-year-old girl with sickle cell disease, and you can see this whole region of the brain is not getting blood, not getting oxygen. But the other stat that's not here is that 30 to 35% of children with sickle cell will have a silent stroke. That's a stroke in a cognitive part of the brain that doesn't present itself physically, so the kids might start getting bad grades in school, not thinking properly, and they go to an MRI and find all these stroke lesions in the thinking portions. So this is a very big problem, and there's no real cure for it, of course, because it's a genetic disease. But Bob Adams led some amazing studies back in the 90s into the early 2000s, where he actually found that doing transcranial Doppler to measure the centerline velocity of the middle cerebral artery in children with sickle cell disease was actually indicative of those that were at greatest risk for strokes. So if that centerline velocity was greater than 200 centimeters per second, then that child was at risk for stroke. And what they would then do is put that child on a monthly blood transfusion regimen, because that was the only thing the only thing you can do. So monthly blood transfusions from now until I think it's the late teens when they might stop. And this is still the only treatment for it. But being the engineer that I am, or as I was trained, well, if you have an increase in centerline velocity, usually that's an indicator of some sort of stenosis or something else that might be happening. And again, these strokes are large artery oxygenated blood diseases. So we began to start looking at it. And I take you back to some of my training back in my PhD time, where my background was in shear stress, endothelial cell biology, and vascular remodeling. And we all know that in the carotid artery, you get at this bifurcation, you get this region of flow separation that leads to oscillatory shear stress on the outside wall. That's been correlated with regions where plaques will form inside of the artery wall. And there's been many studies about all of the different endothelial cell changes in response to that disturbed flow that causes pro-inflammatory, pro-thrombotic, and pro-remodeling reactive oxygen species environment. And what I began to study, again, during my dissertation work, was how these proteases called cathepsins, they're a family of cysteine proteases, are actually also regulated by shear stress in endothelial cells, where in the straight parts of your artery where you have unidirectional Poussey flow, these are actually turned all the way down. We don't find their expression at all, and the endothelial cells actually produce the inhibitor in large amounts. However, under sites of disturbed flow, everything switches. They start to produce the proteases and downregulate the inhibitor. And these proteases are actually very powerful elastases and collagenases that can remodel the major structures in the artery wall and promote these plaques to form. Now, why it's interesting is in sickle cell, one of the reasons to believe that these arteries close off is due to vascular remodeling, due to smooth muscle cell and neointimal formation in the cerebral arteries. But the mechanisms by which this is happening are not really clear. Now, there are added problems with sickle cell is that when these cells break open and lyse due to that sickling, the sickling of the hemoglobin, they release the hemoglobin from the red blood cells, which actually scavenges nitric oxide. So normally under high shear, endothelial cells make more nitric oxide. It goes to the smooth muscle cells. They relax, and the vessel opens. Because the nitric oxide is not reaching smooth muscle cells, there's no vasodilation, so you have the vessels getting smaller. The smooth muscle cells are proliferating to now add cellularity to that lumen narrowing, and then ultimately you'll get a blockage and a stroke. But again, many of the causes, the other causes are not clear, but there's increased red cell adhesion, monocyte adhesion, increased TNF-alpha, and we'll talk about how we use those to understand why these kids are having strokes. So again, I mentioned this already. During my dissertation work, we understood the shear stress regulation of these proteases, and now in my own lab, starting in 2009, we're trying to develop this personalized model. How can we predict which patients are at risk for stroke? As I mentioned, 11% will get strokes. Doctors don't know how you determine which ones for any reason except wait until the pathology has begun with the transcranial Doppler. So our idea is to do this multiscale model. Can we start with the enzymatic activity, these cathepsins in the vascular wall that we know remodel that artery? How are these regulated by cells? We know these endothelial cells respond to both biochemical insult as well as biomechanics related to the shear stress and flow environments. Then how does that play together to remodel the tissue and the structure of that artery, then to close that artery off? And along the way, will we find any biomarkers we can measure or any other druggable targets? Because as many of you may know, sickle cell, we're now, I think, 102nd year of knowing about the disease, and there's one drug on the market still, right? So there still is plenty of opportunity for that. And this work, I have to brag, I got my new innovator for, so nobody else was funding sickle cell, so I'm thankful to NIH for doing that. But a little bit of detail about these proteases. Again, these are also a little outlier class. A lot of people like MMPs when they study proteolytic remodeling in tissue. But the cysteine proteases, they were first identified in lysosomes. You know, lysosomes are your trash compactors. They degrade all the proteins in your body. So these are very powerful, but they've been shown to be secreted by osteoclasts, macrophages, and other cell types. And we know macrophages enter that vascular wall and help advance that plaque. They are also the most powerful human elastases and collagenases. So cathepsin K, one we'll talk about quite a bit, is used by osteoclasts to resorb bone, right? That's the key enzyme for doing that. They're regulated very heavily by the body, being synthesized as inactive precursors, plus they prefer an acidic environment, acidic pH, and a reducing environment because they're in lysosomes. So it used to be thought way back in the day, well, if they get out, they don't work because your body's neutral outside of the cell. But we all know that under certain disease states, this is actually not true. So they're very sensitive because the body regulates them so well during, it makes for a tough PhD when you can't find the molecule you're looking for. Though you know it's there, very tough. So, my PhD advisor, he was trained as a physiologist, and so, I took all these great classes on math modeling and said, we should model it. And I just did the experiment. Okay, so then it was tough. So then I said, okay, come back to my own group. We're going to do the math model because I think that's going to add a lot more information that we can't get experimentally because they're quite transient molecules. So what we wanted to know is, can we develop a model of how the cell might release these proteases that bind the substrate and degrade it? And again, they could release it in the endothelial cell into the bloodstream, which maybe doesn't have an effect on the wall, or release it basally, where then it can interact with all the matrix in the wall. And I take you back to your old school kinetics, where there's a proenzyme that would be released. There's some rate of activation for it to the active form. When it's released, it can bind to that inhibitor that I said many cells make, which takes it to an inactive complex. Or it can bind to the substrate, forming a complex where there's some rate of catalysis and release the product. Or again, when it goes back to that mature enzyme, there's also some rate of inactivation or degradation just due to the conditions, right? So we could combine these, again, so a lot of biochemical studies have done very well for the binding and catalytic rates, but the rates of activation and degradation are very much dependent on the environment and the conditions. So again, we can write these governing equations, the system of differential equations, and solve it simultaneously. And first we use a synthetic substrate just to make sure we got the right fit for the one enzyme. So I'm going to tell you how we'll expand on this in 1 second. But the other thing that I did to save my graduate students time that they should be thanking me for still is I knew that we needed assays with increased sensitivity. The old ways it was had to do 3 different assays, 3 different substrates. These enzymes, because they are very conserved family, they like the same substrates. There's promiscuity, just with different. It just gets to be a lot of experiments that reviewers ask. And so I developed modified what people normally have heard for MMPs. This is zymography, where you can do proteolytic activity. But these don't work under the same conditions that MMPs work. So we then generated conditions to get their activity. So again, same principles. We made a polyacrylamide gel impregnated with gelatin, which is a substrate that all of these enzymes can degrade. You can lyse your cells, put them in this buffer, just like you're doing a Western, except there's no beta-mercaptoethanol because we're not breaking disulfide bonds. We can run the electrophoresis to separate them by their molecular weight. And then instead of transferring them to a membrane for a Western, we're actually going to wash out the SDS, which will renature these enzymes into their thermodynamically favorable conformation in the gel at the site where they were separated. Then you can add them to assay buffer, perfect for their activity. So low pH, reducing environment, 37 degrees, incubate overnight, and they will actually cleave that substrate, that gelatin substrate, at the site at which they were. So when you stain with Kumasi blue, because there's protein in the entire gel, it all turns blue, but you get white bands where the enzymes were active. So what we actually found out from this is that this actually, for cathepsin K, this very powerful collagenase and elastase, was much more sensitive than Western blots. You can see with the best antibody for CAT-K on the market, we lose that signal around 1 to 5 nanograms, but we can detect down to 0.1 nanograms with zymography. And this is also only detecting the active enzyme, not the inactive form, because it has to be able to cleave this gelatin substrate. Then, because we're engineers, we loved it. We actually were able to multiplex it, and we can detect 4 of this family of enzymes all at once. And you can see they migrate to different sizes in this electrophoresis, so we can uniquely identify them, all from one tissue or lysate. And again, this is also nice as a PI. There's no antibodies, so it actually is very, very, very cheap. So here's back to the big goal. We want to then combine this kinetic network of these 4 enzymes, and we actually are doing V now for some specific reasons, with the rates of their activation, their degradation, but again, they all bind to these different matrices, all at different binding affinities. They catalyze at different affinities, and this is the full-scale model that we're trying to create. So then we tried to do the experiments with all 4 enzymes. It got really crazy. So we backed it out and said, let's just do 2, and just 2 substrates. And this is where the modeling comes in, and I'll relate it back to sickle cell and by the end. So there's this thought in the protease field that if you have more proteases, you'll have more product form, right? That seems to make sense. If you have more things to cleave the substrate, you'll have more. So we said, well, we have K all by itself degrading elastin, and we know the endothelial cells secrete K, and they also secrete S at the sites of disturbed flow. So K degrades elastin this much, S this much. So we put that into our kinetic model and said, well, if we put them together in a tube, you should get this additive effect, right? We get this much elastin degraded, particularly since the substrate is not limiting. It's a huge excess of substrate. But when we actually ran the experiment, we only got a slight increase in the amount of elastin degraded. And this puzzled us for a very long time. But then we began to think about what happened. So we ran some Western blots after this experiment. If we were to incubate the two together, we saw that S stayed around for the entire 2-hour period, but cathepsin K was gone by that 30-minute period. Then we said, well, what happens if we run K by itself? And K still degrades itself, but at least it can make it till 60 minutes. And this led us to think, is S actually degrading the cathepsin K in the system? And so we came up with this term, cathepsin cannibalism. All right? I love this term. Very exciting moment. And it actually did all the discrepancies. So we defined cannibalism as when one cathepsin eats another instead of substrate. So you can see here in this kinetic analysis, there's less free enzyme to bind the substrate. Plus, when you degrade that out of commission, then there's much less overall. So we had to change our model architecture to include the S eating K terms here, where you get now cathepsin K degraded from S. And when we do that, again, here's our inert model where we expected this additive effect. We fit our parameters and actually get a much better fit. So, okay, no model is real. I always tell my students, no model is real unless you test it, so no offense to any modelers who don't do experiments with it. But we can do both, so that's why I say it. But we looked, well, are there matrix-dependent effects? Because what's different, cathepsin S and K are both very strong elastases. But K is a really strong collagenase as well, and S cannot degrade collagen very well. So we said, well, if S eats K, can you increase the ratio of one to the other and protect the substrate by taking out the predatory cathepsin? And so we first modeled it, and we said, well, if you increase to a 10-fold ratio, you can reduce how much collagen is degraded and take out all the K. And then we went on ahead and tested it. And so again, you can see here with the Western blot, as we increase the amount of S to K ratio, at the 10-fold ratio, you see a significant drop in the cathepsin K that's present. And then when we run the Zymo, again, we can detect both. You see at the 10-fold where the K drops, it goes away, but you still have the S. And then here's the key experiment. When we did the collagen 1 degradation, you see there's the greatest amount of collagen remaining when you have this 10-fold ratio of S to K. I'm sorry, I'm going to highlight it. So again, we can protect the collagen 1 by using one enzyme to actually eat the other. This is exciting. But then we put these constants back in our model and said, well, if we increase it, you can see, again, by that 10-fold ratio, we can protect collagen. But what happens with elastin? Because they both degrade elastin. So again, what's good for the goose isn't necessarily good for the gander. Because they both degrade elastin, despite cannibalism, as you increase S, you will still get a reduction in the amount of elastin in that tissue. Now, why is that important? And so we published that in JBC last summer, and that's my student Zach, who did a lot of the work. He's fantastic. But it takes us back to the sickle cell artery. In the arteries, there's lots of elastin, which is a key protein that when it gets degraded, smooth muscle cell proliferation and migration happens. But there's also collagen present in these lamellar units as well. So as these cells are releasing both of these, how much of this matrix is actually getting degraded? And these are things that we now have to start considering. But the other question is, well, are cathepsins being upregulated by sickle cell disease? So we came back to using, again, monocytes binding to endothelial cells. Again, many of you know, under disturbed flow, endothelial cells express ICAM1, VCAM1, monocytes bind, and then enter the vascular wall. But the complicating factor of people with sickle cell is the chronic inflammation leads to increased TNF-alpha, which TNF-alpha also increases ICAM1 expression on endothelial cells and increased monocyte binding. So we first tested this with THP1 cells and looked at if direct contact or indirect contact would increase cathepsin activity in endothelial cell monocyte cultures. And we actually found out, very interesting, that with TNF-alpha, it would turn on cathepsin K in the endothelial cells. Monocyte binding, not so much. Monocyte binding would increase V, but you could see that K was being turned on in the THP1s. And this was also very much a direct contact mediated phenomena, because when we separated them, there's some effect through paracrine factors, but the direct contact was much greater. So now that was with the cell line. But we then wanted to know, well, what happens when we actually get PBMCs from people living with sickle cell disease? So we partnered with the Sickle Foundation of Georgia to get blood, and then we isolate the PBMCs from the blood, and then we co-culture those with the endothelial cells in a direct contact state. And what, again, this is what other people have shown already, that there's increased adhesion of peripheral blood mononuclear cells from people with sickle, and you can see the difference here. We also made sure that the TNF-alpha was elevated in the sickle cell blood versus the wild type. And again, here's a picture of the monocytes adhered to the endothelial cells. But what we saw was actually so surprising. So we couldn't get the wild type PBMCs to even adhere without TNF-alpha. So we stimulated those with TNF-alpha. And again, we can get cathepsin K, but with the sickle PBMCs, without any exogenous TNF-alpha stimulation at all, they already bind to endothelial cells and turn on this powerful protease. So again, this is a powerful elastase and collagenase that has been implicated in cardiovascular remodeling. So it's almost like they don't have a chance. They have increased TNF-alpha, increased monocytes, and then all of that will bind and turn up this protease automatically. So we wanted to look at the mechanism by how it does that. And so we used Bioplex, which are these phosphoprotein beads, to look at which point is which are the signaling kinases that are turned up in response to either TNF or monocyte binding. And you can see the key one that seemed to be responsive was J and K and its downstream target of c-Jun. So then we went back, got new blood samples from sickle cell individuals, and decided if we use a J and K inhibitor, can we block that upregulation of cathepsin K after the monocytes have adhered? And we were very excited to see it. So again, this is just ECs with TNF. You can see J and K reduces that signal here. But when we look at the sickle PBMCs, no TNF, again, you turn on K by themselves, but when we add the J and K inhibitor, we can reduce that signal by 50%. And as I mentioned, there's no drugs on the market, and so maybe this is another path to pursue. Now, of course, J and K is in lots of signaling pathways, so there's some things that are working out there. But this is actually very exciting for us. And so now we take it back to now, that was all static culture, but as I mentioned, what got us thinking is this disturbed flow in sickle cell disease. And again, when we're seeing this centerline velocity increase. Now we know if there's some aggregation or luminal narrowing, downstream of that, you're going to get post-stenotic flow and flow separation and reversal. So the hypothesis then was in a person with sickle, since there's going to be aggregations throughout the vascular tree, is it possible that they have more regions of disturbed flow throughout their arterial tree? And so the way we model that is we use a cone and plate apparatus. We can grow a monolayer of endothelial cells, put the cone on top, and with the motor, we program it to rotate either in one direction, such as you'd find in the straight parts of your vessel, or an oscillating direction, such as those sites prone to vascular remodeling. And here's a nice image of it. And this actually, it's pulsatile to fit with the systole and diastole. This is from the Gimbroni group. They've defined these profiles, and these are actual cones here. And we can run those for 24 hours. And again, what we find is my old work, though, but this is new stuff. But again, with the pro-remodeling shear, you turn up cathepsin K, but with TNF-alpha, of course, you keep that turned up, all right? And again, with J and K, it's turned up, as we showed before, and also downstream c-Jun. This is not a disturbed flow. But what's interesting, in the vasoprotective blood flow, the unidirection, what would be in the straight parts of the arteries, even with TNF-alpha stimulation, the endothelial cells do not express cathepsin K. So now there's this balance, or this conflict between the biomechanics and the biochemical signals that the cells are resolving and making a decision, even though J and K is still activated. So even though the mechanism we used before to block it, J and K and c-Jun are both activated and elevated under vasoprotective shear, but cathepsin K is not being produced. So now we're looking at what are these conflicting statements here. And again, we did the mRNA to show also that the gene itself is not even being turned on. So we're doing some chip analysis to see if that's still binding or not. But so altogether, to put that together, we're looking at this large artery remodeling in sickle cell and how it arises from this dynamic interplay between the biomechanics and the biochemical signals. And again, under vasoprotective shear stress, we're able to block K, although we're still activating J and K and c-Jun. But under the pro-remodeling, everything works the way we thought it to work previously. And so how we're now putting that together, we're collaborating with a computational fluid mechanician over at Emory, who's in the math department there, where we're actually getting MRAs or angiograms from patients. And he's actually able to recapitulate the geometry so that we can then identify the shear stress and the fluid dynamics in a patient-specific fashion that we can then program into our cone and plate shear apparatus. So we can now do kind of some patient-specific fluid profiles and look at their upregulation of these enzymes with and without the sickle red cells as well. And again, this is the whole picture we're trying to put together. So one of the challenges that came up in our session this morning about linking one step to another, and that's where we're trying to meet with this multiscale approach, although we're going to come across a few different challenges. But the idea here is, can we find a way to make it more personalized and patient-specific instead of just having this one assay? So I want to thank my group. I want to thank you all for listening. And this is us. Actually, we did the sickle cell run. It was a 7K to raise money for the Sickle Foundation of Georgia. And they all came out, and I did run it as well. But I didn't beat all the young people. But that's fine. Some of them did walk. But again, thank you for your time. Over to questions.